ओके, ऑन द 3 प्राइम एंड देयर इज अ पॉसिबिलिटी कि जब हम न्यूक्लियोटाइड डालेंगे तो इस 3 प्राइम एंड पे भी इनकॉर्पोरेशन हो सकती है। ओके, टू प्रिवेंट दैट, कैन यू सी ब्लू कलर है यहाँ पर, उसके बाद एक रेड है, ब्लू कलर है, रेड है। So we have deliberately added dideoxy ATP at the 3 prime end so that the polymerization does not take place at the 3 prime end. So this is double ensured. तो हमारा जो dideoxy ATP है वो दो काम कर रहे हैं। एक तो वो बाइटन लेबल्ड है, बाइटन लेबल से वो इट इज हेल्पिंग इन द प्यूरिफिकेशन एंड द एनरिचमेंट ऑफ द लाइब्रेरी, एंड सेकंड इज दैट इट इज ब्लॉकिंग द 3 prime end so that no nucleotide incorporation can take place at the 3 prime end. Okay, I hope it is understandable. Now, once you have loaded the library, the next step is to add the nucleotides. So you generally add 4 nucleotides, that is A, T, C, G, to the sequencing chamber. And once you add the ACTG to the sequencing chamber, then there is a so this is how the whole, this is now I'm talking about sequencing. So this is how the process of sequencing takes place. So if you see over here, this is a polyadenylated tail, and this is polyadenylated tail has been attached complementary to that of the T oligos which are present on the surface of the flow cell. So then this modified nucleotides one at a time, remember कैसे जाते हैं, one at a time, इसको हम कहते हैं कि the sequencing is performed in quads. What is quad? Quads means a group of 4. That means a group of 4 cycles, and in each cycle one nucleotide is added at a time. जैसे इस cartoon में you can see that only C is added during this sequencing cycle. That only we are seeing only C, fluorescently labeled C. अब ये जो fluorescently labeled C है बेटा, this is a very important fluorescently labeled C. Why this is a very important fluorescently labeled C? That this fluorescent label is a virtual terminator. What is happening here is that it is attached to an inhibitor, and the inhibitor molecule, so fluorescent label is attached to an inhibitor molecule which does not allow the further polymerization. Okay, so एक molecule यहाँ पर complementary base pairing की help से लग गया. Once it is attached over there, तो third step में क्या होता है, light or the light is thrown on the flow cell. तो उससे क्या है, फ्लोरेसेंस प्रोड्यूस होती है. द फ्लोरेसेंट मॉलिक्यूल व्हिच इज अटैच्ड टू, ये देखो रेड हो गया, इसका मतलब है कि देयर इज फ्लोरेसेंस इज प्रोड्यूस्ड. सो जो फ्लोरेसेंस प्रोड्यूस होती है दैट इज रिकॉर्डेड बाय द डिटेक्टर्स. ओके, नाउ एक्सेस ऑफ आयंस आर रिमूव्ड ड्यूरिंग द वॉशिंग स्टेप. ओके, जब हमने इसकी फ्लोरेसेंस को रिकॉर्ड कर लिया, नो व्हाट डू वी नीड, वी नीड टू नाउ रिमूव दिस मॉलिक्यूल. अब यहाँ पर फर्दर कोई दूसरा मॉलिक्यूल क्यों नहीं आके जुड़ रहा है, क्योंकि क्या लगा हुआ है, इनहिबिटर लगा है. इनहिबिटर एक लिंकर की हेल्प से लिंक है विच इज नॉट अलाउइंग फर्दर पॉलीमराइजेशन. तो अगर यहाँ पर सी होता, यहाँ पर मान लीजिए जी होता, तो दूसरा जी सी आके यहाँ पे नहीं लग सकता है. सो दैट इज द ब्यूटी ऑफ दिस टेक्नोलॉजी. सो वंस द फ्लोरेसेंस हैज बीन रिकॉर्ड हो गई, उसके बाद द नेक्स्ट पार्ट इज दैट सर्टेन enzymes are put into the reaction mixture which cause the cleavage of the linker molecule. जब linker molecule cleave होता है, then fluorescent molecule also goes and the inhibitor also goes. So you will be left with C only, which has been incorporated into the complementary DNA strand and with no fluorescence and no inhibitors, which means now this molecule is now ready for the second cycle, that is the incorporation of the second nucleotide and the second cycle of the helicos sequencing. इसी तरह से मान लीजिए अब G आता है, when the G will come, it will bind to this molecule and it will be fluorophore labeled, and then excess of G fluorescently labeled nucleotide will be washed away, and then there will be imaging method, light source will be put on the fluorophores, they will be accented and the signal will be recorded. Every nucleotide has a characteristic phosphorophore attached to it. Once the fluorescence is recorded or imaged, then again it will be cleaved, leading to the release of the inhibitor and the fluorescent molecule. तो ये चलता रहता है, and this is performed in quart status in the cycle of 4. Okay, till all the 4, and this is repeated again and again. Okay, now the sequencing machine knows how to do this process. Okay, so imaging में क्या होता है बेटा, जो हेलिकॉस सीक्वेंसर है या हेलिकोप सीक्वेंसर है, this heliscope sequencer, it has got a CCD camera and it is able to record the fluorescent signal which is produced by the nucleotide. So you can see that 635 nanometer का laser emit होता है. ठीक है, the laser is there, and when there is emitted fluorescence is there, this fluorescence can be detected by the CCD camera which is present on this side. So this incorporation is recorded very efficiently. And you can see that the flow cell will have thousands of single molecules present on its surface, and the images will be recorded for all these molecules. Every cycle, the image. So these are the images. You can see that these are the images, and so for every image, think about it, every image is recorded, and this is kind of a galaxy here of images that you get once the images are taken by the sequencer. In a standard sequencing run, generally 120 cycle of the individual nucleotides addition are done in a standard helicos run, but it depends. The runs can be increased or they can be decreased depending upon what is the research question in mind for which the helicos sequencing is being done. So once the data is there, recorded by the camera, it is processed, the images are processed, and the digital, that means the camera signals, fluorescent signals, are then converted into the ABCD ACTG signals. That means digital signal को हम एक alphabetical signal में convert करते हैं by using different algorithms, and we won't be discussing about those. Okay, so talking about the through high throughput or the throughputness of the helicos sequencing, तो जब ये शुरू हुआ था बेटा, तो generally read then 35 base pairs थी और around 30 GB of data generate होता था, ठीक है, and generally the runtime was 8 days. So ये थोड़ा सा इसका limitation था कि बहुत लंबा read नहीं मिल रहा था. We were not getting too much lengthy reads comparable to the initial 454 की तरह हमें we were getting the reads for initial Illumina reads. So however, the advantages of this technique was that it was able to solve the problem of homopolymerism, which was there in the case of the specifically the 454 sequencing. ठीक है, तो उस problem को बहुत efficiently solve किया था helicos sequencing में. But at the same time, it did not involve any PCR bias because there was a single molecular sequencing strategy. And the primer dimer formations was not there, and there was a very simplified data analysis. Photo खींचो और उसको analyze करो. But there was a disadvantage. What was the disadvantage? That you have to stop the process of sequencing every time you need to add the nucleotide, because every quad run, it involves the addition of 4 nucleotides. So there was a lot of halt during the sequencing process. And then due to the halt, the time for sequencing was increased to 8 days. So that was quite a long amount of time. Then the read length was also very small, 32 nucleotide. Above this, the only other problem was that there was also high error rate. Why high error rate was that? Again same, अगर आप एक molecule से data लेंगे, there are chances of misincorporations. So that is, data is a little less reliable. But if you have clones, then you can find out that which is the base which is exactly there. So therefore, there was a need to increase the accuracy, there was a need to increase the read length, and therefore also a need to increase the cost per base pair sequencing. So these were the limitations. That is why people did not use Helicos much, and therefore they became bankrupt. Like that of other sequencing technique, it can be used for genomic sequencing, for variation detection, for chip assays, and also for single molecule measurements of the RNAs. Okay, so the applications are of almost, they were the same, but now the sequel, um, it is trying to revive this technology. So
true single molecule sequencing has become a reality. Let's take a closer look at how it works. Sample preparation is simple and can be performed by a single person in less than a day in a tunable microplate. A DNA sample is cut into short strands of 100 to 200 nucleotides. A RNA universal protein sequence is added to the 3' end of each DNA strand. Each strand is labeled with the addition of a fluorescent adenosine nucleotide. These labeled strands will serve as templates for the single molecule sequencing chemistry. The DNA strands are then hybridized to the flow cell, which contains billions of oligo-T universal capture sites that are immobilized onto the flow cell surface. Each individual template that hybridizes to the flow cell surface will generate its own sequencing reaction. Because the Helicos sequencer detects single molecules, the templates can be mapped at a very high density of over 100 million templates per square centimeter, or millions per row. Once the samples have been hybridized to the flow cell surface, they are brought into the instrument. A laser then illuminates the surface of the flow cell, showing only a hint fluorescently labeled template. A 60D camera produces a map of the templates of the flow cell surface. The instrument takes mirror images in a stepwise pattern across the flow cell. After the templates are been linked, the template label is cleaned and washed away. The sequence of reaction begins by introducing a dedicated nucleus and a resident labeled filter, in this case a acid. A global T capture surface serves as sequencing parameters for the PSNS process. DNA organize parallels the addition of the labeled adenosine retrieval particles in a template format. A wash sector removes the fluorines and creating a homographing filters. The fluorines of several molecule templates that have incorporated an A are then pushed along the packaging the entire flow cell surface. After finishing, a package step removes the fluorescent elements. The process continues for each of the remaining bases, C, G, and T, and repeats until the desired value is achieved. Sequence information is collected at each new base edition, billions of strands at a time. Common application of these sequencing technologies, the PSNS process is a sequence, every strand is unique and sequenced independently.
So I hope this video is available in the net also. Kindly go through it if it is not audible over here. Okay.
True sync.
So this is the URL of this video and you can just go through it. So billions of fragments are synthesized independently, but as I have already told, one of the greatest limitation was that it was very expensive, that lot of reagents were used because at one time only one nucleotide is flown through the sequencer. So heavy usage of reagents led to this technology being expensive. So that is why it became obsolete in 2012, but it is still being revived. I hope you like the presentation. And this is how we finish with the sequencing presentations of the different sequencing technologies. I wish you again best of luck. I will be giving you exam of 4 sequencing technologies at one time. Kindly go through it and do respond during the Google dashboard. Thank you so much. You have a great day.